Now I'm going to talk about bats and Hendra virus. As an introduction, the behavioral ecology of many species of bat identifies them as potentially efficient vertebrate disseminator hosts of mammalian viruses. Bats are unique in their response to viral infections in that they can sustain them without developing disease. Evidence of a wide range of diseases have been identified in bat species. Some of these diseases are exotic to Australia, like the severe acute respiratory syndrome, Nipah virus, and Ebola virus, while zoonotic diseases found in Australian bats include Australian bat lyssavirus, Hendra virus, meningovirus, and Leptospira bacteria. Hendra virus is an RNA virus belonging to the family Paramyxoviridae, genus Nipah virus. While serologic evidence of Hendra virus infection has been found in all 4 species of Australian flying foxes, more recent research suggests that 2 species, the black flying fox and spectacled flying fox, are the primary reservoir hosts. Clinical disease due to natural Hendra virus infection has only been observed in horses and humans. Though there have been 2 recorded cases of infection in domestic dogs that were living on properties where equine cases of Hendra virus occurred. Hendra virus cases have only been reported from Australia. However, serological evidence of Hendra virus infection has been reported in 6 species of flying foxes from Papua New Guinea. It is possible that these reports reflect cross-neutralization with Nipah virus or an unidentified Nipah virus. Related Nipah viruses have been found in bats in Asia, China, and Africa. History. In September 1994, a sudden outbreak of an acute respiratory syndrome resulted in an immediate shutdown of the horse racing industry in southeast Queensland. During the outbreak, 13 of the 20 infected race horses died. The syndrome was characterized by severe respiratory signs and high mortality. The causal agent was unknown. Quarantine procedures and movement restrictions were immediately put in place, and epidemiological investigations were commenced. After the outbreak the putative causal agent was identified. The virus, a previously undescribed member of the family Paramyxoviridae, was initially named equine morbillivirus, but was subsequently renamed Hendra virus, after the Brisbane suburb where the outbreak occurred. The new virus demonstrated its zoonotic capability when a trainer and a stable hand that were in close contact with an infected horse became ill with severe influenza-like symptoms. The trainer was hospitalized and subsequently died after respiratory and renal failure. Infection with Hendra virus was demonstrated in both human cases. A second outbreak occurred in October 1995, after the death of a stud owner who suffered a relapsing encephalitis disease was attributed to Hendra virus. This outbreak resulted in the death of 2 horses and a farm in central Queensland. After extensive investigation, antibodies of Hendra virus were not detected in domestic animals, so it was determined that Hendra virus infection was not highly contagious in horses, and that direct contact or mechanical transmission of infectious body fluids was necessary for natural transmission to occur. It was until September 1996 that a Hendra-like virus was isolated from the reproductive tract of an apparently healthy gray-headed flying fox. In comparison of the bat isolate with the isolate from horses, showed it to be indistinguishable from Hendra virus by a range of tests. In addition, in a retrospective serological survey of Australian flying foxes, antibodies for Hendra virus have been identified in sera collected in 1982, which is the earliest sample tested. In January 1999, a new fatal case of Hendra virus infection was reported in a horse near Cairns in North Queensland. However, a companion horse was unaffected on clinical or serological examination. Impact. The impact of Hendra virus infection on flying fox populations is presumed to be minimal, as it seems to be subclinical in this species, making them natural hosts of the virus. In horses, acute clinical signs include pyrexia, increased heart rate, dyspnea, sometimes with frothy nasal discharge, and rapid progression to death associated with either respiratory or neurological signs. Colic and acute death have also been reported. Morbidity may be up to 100%, while fatality rate has been estimated at 90%. Humans can develop pyrexia, neurological or respiratory signs and symptoms. Of 7 reported human cases, 4 have resulted in death. Epidemiology. Serological evidence of Hendra virus exposure has been reported from flying foxes from Western Australia, the Northern Territory, Queensland, and New South Wales. Black and spectacled flying foxes appear to be the primary reservoir hosts for the virus and the source of infection in horses. There is evidence that infections occur sporadically in flying foxes across Australia. Latent infection with recurrence may be an important epidemiological factor in infection in flying foxes, whose urine is believed to be the most important route of excretion and transmission of the virus. Viral infection and shedding fluctuates, with a short viremic period and small numbers of flying foxes actively infected at one time. Viral transmission between bats appears efficient. Seroprevalence is high and widely distributed in flying fox colonies. Hendra virus spillover from flying foxes to horses likely has complex causality involving multiple host and environmental factors. Paddock house horses are most at risk and are likely exposed when they stand underneath fruiting or flowering shade trees in which flying foxes are feeding or roosting, or have recently visited. The most likely pathway of transmission is when infectious urine, body fluids, or excretions of flying foxes come into contact with a horse's mucous membranes. This may occur directly or indirectly, the latter via contaminated pasture, feed, or water. The incubation period for horses is believed to be 5 to 16 days. Horses may be able to shed virus for 2 days prior to the onset of clinical signs. Close contact between horses or between horse and human appears necessary for spread of infection. Viral loads in acutely infected horses are usually very high, but horse-to-human transmission is considered to be inefficient. So, procedures undertaken by affected veterinarians and assistants may have significantly increased their exposure to the virus. The reported incubation period in humans vary between 5 to 21 days. There is no evidence of a spread of Hendra virus from flying foxes to any species other than horses, and no evidence of human-to-human spread. Other mammals, including dogs, may become infected following exposure to viremic horses, becoming a potential risk of transmission to humans. While there is evidence of recurrence of Hendra virus infection in humans, experimental studies indicate that virus persistent in convalescent animals is not likely to be a risk for transmission to humans. Multiple hot spots of Hendra virus infection in horses are found along the east coast of Australia, with the largest hotspot extending from southern Queensland to northern New South Wales. The geographic spread of Hendra virus outbreaks in horses falls entirely within the geographic range of the species P. electo, and most events usually occur in winter, in the southernmost part of the distribution of P. electa. The winter seasonality of echoing Hendra virus cases is mirrored by a strong seasonality in Hendra virus expression in pooled flying fox urine samples, which may have increased survival at cooler temperatures. Ecological change seems to be the most plausibly key factor in the emergence of Hendra virus. Available data on many fruit bat species suggest that populations in Australia are in decline and disruption throughout their range. Anthropogenic activities, primarily habitat loss and hunting, have been identified as constituting the major threats. Deforestation, whether for agricultural land, commercial lodging, or urban development, is widespread in the region and results in loss of abandonment of roosting sites and the loss of feeding habitats. Secondarily, habitat loss due to clearing is commonly intensified by tropical storms, the remnant forest being particularly prone to high wind damage. Hunting, whether for consumption or crop protection and at both a local and at a commercial level, results in the abandonment of roost and feeding sites. A scenario emerges of fruit bat populations under stress, of altered foraging and behavioral patterns, of niche expansion, and of closer proximity to man. In Australia, the geographic redistribution of roosting sites has been increasingly into urban areas in recent decades. Control. Prevention of the infection in horses and humans is aided by the use of vaccination in horses, limiting exposure of horses and their feed to flying fox contamination, and by use of appropriate personal protective equipment, particularly when dealing with sick horses and undertaking post-mortem examination of suspect horse cases. Post-mortem examination is believed to carry the greatest risk, and some potential transmission risks exist prior to the onset of clinical signs in horses. Surveillance. At the national level, health matters are mainly regulated and managed by the Department of Health for human aspects and the Department of Agriculture for domestic animals and wildlife health. State governments also have a leading role in managing health issues. Hendra virus research is coordinated nationally through the Intergovernmental Hendra Virus Task Force. The task force is made up of the respective heads of biosecurity agencies in Queensland, New South Wales, Victoria, and the Australian government, chief veterinary officers, chief health officers, chief scientists, and senior environmental representatives from Queensland and New South Wales, as well as the Australian chief veterinary officer and head of the Australian Animal Health Laboratory, CSIRO. The Auswed Plan Response Policy Brief for Hendra virus infection details the national response policy for eradication of Hendra virus infection in terrestrial animals. Hendra virus is not an OIE listed disease. As conclusion, with these two diseases, the importance of the growing contact that occurs simultaneously between domestic animals, wildlife, and human population, enhanced by the diffusion that these agents may suffer thanks to globalization, is revealed. In order to monitor emerging diseases, it is necessary to have multidisciplinary work teams with professionals trained in medicine, epidemiology, virology, zoology, and veterinary medicine, due to the great interrelation of factors to which these diseases are subject. Finally, it is important to promote the development of research teams that work with risk analysis systems and look for potentially transmitting species of pathogens in order to understand and therefore control the evolution of emerging diseases. Thank you very much.